Great. Our next speaker is Wenbiao Chen, and he will speak about overnutrition induces macrophage and Ripk3-dependent beta cell death in zebrafish with muscle insulin resistance. Thank you.
Thanks, Nils. I'd like to thank the organizer to give me the chance to talk about our effort in the last few years trying to model type 2 diabetes pathogenesis in zebrafish. Okay. So, as most of you know, that type 2 diabetes has increased over the last couple of decades, both in incidence and in prevalence. And this is mostly due to the increase of obesity. And obesity causes type 2 diabetes in a couple of ways. One is that mostly it's because it creates an insulin-resistant environment by for example, increase in the low-grade inflammation and the pro-inflammatory cytokines interfere with insulin signaling. In addition, it also increases your blood fatty acid that also can be uptaken, but its metabolites can also interfere with insulin signaling. So this insulin resistance, create a burden to the insulin-producing beta cells, so they have to compensate it. They usually compensate by either increasing the insulin secretion and also insulin the beta cell number. And so this just a nomenclature is in the field, and this early phase is called beta cell compensation, and afterwards beta cell dysfunction and then beta cell failure. The mechanism underlying this process is not very well understood. So we, a few years ago, tried to use Zebrafish larvae trying to model this process and trying to understand its molecular mechanism. So to do that, we need to first create a transgenic line that marks the individual beta cells. And we decided to use a nuclear localized beta cell line because the beta cells are clumped into a very small space. If you use cytoplasmic, it's very hard to distinguish one to another. And the advantage we're using the larvae is because there is only one islet at this stage, and they're very easy to count because there are about 30 to 35 beta cells at this stage. And it's also that because they start to feed. So we can manipulate the insulin demand by manipulating the nutrition. And we can also manipulate insulin demand by drugs and also by transgenesis. And we focused on one model which is inspired by Steve Faber's work that he actually told my postdoc that a fish, the larvae loves the egg yolk very much. They just can't stop eating it. So we thought that's a very good model to increase overnutrition. So we started a culture of the 60 larva fish into the chicken egg yolk 5%. We usually prepare them fresh, and we find that you have to culture them for 8 hours to induce a compensatory response. And the response is very robust. Not as robust as you would like, but it's a 20 to 30% increase. Within 8 hours, we thought that's a very good model to start to work with them. And this increase is due to beta cell differentiation from progenitor cells. So using a number of approaches, including drug screen and the transgenesis and gene knockouts approach, we published our study on the mechanism of compensation just recently. And just to summarize it here, so we've found that this process requires the insulin secretion apparatus that is usually induced by nutrients through the ATP-sensitive KATP channel. But if you constantly stimulate this process, because when you eat, it's only just 30 minutes. This process is gone. But if you stimulate for 8 hours, then that somehow induces ER stress and maybe even oxidative stress. We have evidence for ER stress. We are still working on other mechanisms. And those stress induces secretion, non-conventional secretion of FGF1, which then stimulate the differentiation of beta cells. So that was the compensation part was easy to model, but we had a hard time trying to model the necrophase of this process. So what we have to do is we find that we have to combine both a genetic predisposition and overnutrition to create this beta cell, the natal phase of beta cell phenophase of the pathogenesis. That is, we generate a transgenic line that expressed in the skeletal muscle a insulin-like receptor 1 dominant active construct, which blocks both insulin and IGF-1 signaling in the muscle. They are muscle insulin resistant. And if you treat those fish in the same way, but have to do it for 3 days, so 8 hours in the daylight, and put them into nutrient-free conditions overnight and do that 3 days. And by the night of the 3 days, we see, over this course, we see this compensation and the beta cell phenotype very nicely. So you can see the initial compensation, the second day still compensate, and the third day still compensate. But for whatever reason, we find that during the night of the fourth, this nutrient-free period, the beta cell just disappear. It's almost 40% of the beta cells are gone. And if my poster did a heroic experiment trying to monitor this, just remember that this is probably 4 o'clock in the afternoon. She has to do that for another 16 hours every hour to monitor the beta cells. But what she found is interesting, that there seems to be 2 phases of beta cell decline. There is an initial phase, maybe around 8 hours after, but there's a very small decline. But after 10 hours to 15 hours, most of the beta cells are gone during that period. So this is also this decline is also consequential, because in this model we call Zebrafish insulin resistant ZMIR model, that there is an increase of free glucose, suggesting that they are hyperglycemic. So we just trying to cut the story short. So we also have a lot of data showing that we can treat these fish with diabetic drug, both with, for example, metformin and TZD. And we also try to figure out what happened to the beta cells. And we initially, everybody said it's either dedifferentiation or apoptosis, but we have evidence seeing that it's not those. But instead, we had to explore other alternatives. We explored necroptosis, and because we found that it's not apoptosis. So necroptosis is alternative pathway to apoptosis. It's programmed necroptosis, and it's dependent on the activation of Ripk3 or receptor interacting protein kinase 1, and when it's freed from this complex, for example, when you knock out caspase-8, then it can interact with Ripk3, and this activation induces colostrum, and that also activates its downstream target, MLKL, or mixed lineage kinase-like protein, which forms an oligomer. I think it's 6 or 6-mer, that will translocate to the membrane and create membrane channels, so everything from the cell leaks out. So as a consequence, compared to apoptosis, which includes everything within the membrane, and necroptosis leaks a lot of damage the molecular pattern out to the surroundings, so it's thought to be pro-inflammatory. So to test if this is the process, we tried 3 inhibitors that inhibit necroptosis. And of all the 3 compounds can protect beta cells in the ZMR fish, suggesting that at least this pathway may be involved. In addition, we also test this genetically by locking out the critical component of this pathway, the Ripk3, using CRISPR. So this is just a transient lockout, so we call it lockdowns. By injecting the guide RNA into the founder and then score the phenotype in the injected fish. And we show that knockout or knockdown of Ripk3 can protect the beta cells. And we can also show you that this knockout is very efficient. So we usually test maybe 2 to 3 different guide RNA used. And we also design the guide RNA against the functionally important domain. So try to use the most effective one in those kind of experiments. And we're still waiting for the germinal results. So this suggests maybe beta cell dies through necroptosis. But necroptosis is, there is no marker for it, so it's very hard to detect. The only way to detect it nowadays is just through EM, but we had a hard time to find the islet. It's so small place. So we decided to maybe assume that's the case, then what happens? So one of the prediction would be that we'll have inflammation in the islet. So we test that, and we found that there is no neutrophil infiltration in the islet, but there is a macrophage infiltration just in the mutant, in the transgenic fish, and around the time of beta cell disappearance. And then this macrophage comes here and usually leaves the hole after they're gone within 30 minutes. So just another panel picture showing that we think this is a macrophage. They are phagocytosed beta cells because you can see that macrophage can surround the beta cells, and this one is even clearly showing that the process is surrounding the beta cells. So then if macrophage there is a macrophage there to kill the cells, or they are just, or they're just cleaning up the mess. So we're trying to see that. So first we did an experiment trying to kill the macrophage using the classrinate liposome, which can be perfunctionally taken up by macrophage, thus kill them, and that can protect the beta cells from dying in this transgenic fish. And then we used another approach using the mutant line generated by Will Talbot's group, the IRF8 mutant, which ablate the embryonic macrophagegenesis. And so in this line, at the stage we are working with, there is very few macrophage in the fish. And if we, in this mutant background, we have normal compensations and normal development beta cells. However, we lost this overnutrition-induced beta cell loss, so suggesting that, again, macrophage is essential for the death of beta cells. And this is also interestingly that if the macrophage is gone, the beta cell seems to be working fine, and it's because the glucose seems to be okay. So suggesting to us that macrophage is both necessary for the beta cell dysfunction or lower its function, and also for beta cell death. Then how did macrophage get there? So initially, remember there are the two phases of beta cell decline. We thought maybe that the first phase was the necroptosis, which released those pro-inflammatory molecules to attract the macrophage. So we thought if that's the case, we can maybe inhibit necroptosis, and that should block macrophage infiltration. But our results shows that it's not the case. So we used the Ripk3 inhibitor that did not change the macrophage infiltration at all, suggesting that something else is attracting macrophage to the islet. So we did a survey of pro-inflammatory cytokine in the islet from isolated islet cells. And we found that just at the time when we're taking the fish out from the overnutrition at the end of the third session, we didn't see any change in those molecules. But around 8 hours after taking them out, And we saw an increase of both IL-1 beta and IL-8 expression. And knocking them out, again, using the CRISPR approach can result in protection of those molecules. So in summary, I told you that, okay, so my time seems to be up. So this is our working model. So there are still quite a few questions we don't really know. So first of all, we really don't have evidence to show that beta cells are dying through necroptosis. We are making marker lines for necroptosis. Hopefully we'll have answer for that very soon. In addition, we don't know where Ripk3 is working. Is it involved in macrophage activation, or is it in necroptosis, or maybe in both? So with that, I'll take your questions. Okay, very clear. Thank you.
Thank you very much.